This is Martin. He is suffering from a respiratory tract infection caused by a bacterium. His clinician has unsuccessfully tried to cure him using several different types of antibiotics. The bacterium responsible for his lung infection has become resistant to the most widely used antibiotics available. Like Martin, millions of people are infected each year with bacteria that are resistant to antibiotics. Antibiotic resistance is now so widespread that the World Health Organization regards it as one of the major current global health crises. In 2014, the European Commission started funding 15 international research projects to help solve the problem. Among these projects is PneumoNP. PneumoNP gathers together 11 partners from 6 European member states. The consortium comprises research institutes, universities, small and medium businesses. The partners collaborated from 2014 to 2017 to develop a novel solution to diagnose and treat infections caused by antibiotic-resistant bacteria. The Dutch company PathoFinder developed a diagnosis kit to identify the bacteria causing infections within a single sample. This multiplex kit relies on so-called PCR technology. The kit screens for more than 25 of the most important antibiotic resistance genes, including the carbapenem resistance genes, extended-spectrum beta-lactamase genes, plasmid-mediated cephalophorinase genes, and plasmid-mediated colistin resistance genes. The multiplex kit entered clinical validation in 2017. This kit runs on standard laboratory equipment. This makes the kit relatively cheap to use. Using the PathoFinder kit, Martin's doctor can identify whether he is infected with antibiotic-resistant bacteria and prescribe the proper treatment. Sometimes bacteria are extremely resistant to antibiotics, meaning that no antibiotic is available to treat infections caused by these bacteria. That is why we need new antibiotics. Therefore, PneumoNP research involved businesses that are developing new antibiotics. The Italian biotechnology company Cetlance explores the antibacterial properties of the M33 peptide. The Italian researchers developed a specific form of the molecule and tested its efficiency in an animal model. M33 proved effective against infections due to the very resistant bacteria Pseudomonas aeruginosa and Klebsiella pneumoniae. PneumoNP researchers also studied the AA139 molecule, a promising antimicrobial peptide developed by the Danish biotech company Adenium. The PneumoNP team yearned for more. They wanted to provide a more efficient antibiotic formulation that is also safer for the patient. Therefore, PneumoNP studied the efficiency of coupling the new antibiotics M33 and AA139 to nanocarriers. The PneumoNP team developed several nanocarriers using either lipid nanoparticles developed at Utrecht University in the Netherlands or polymer nanoparticles developed by the research centre Ciditec. As proof of concept, the new antibiotics in nanocarrier form were tested against an antibiotic-resistant bacterium associated with serious respiratory tract infections, the Klebsiella pneumoniae. Fraunhofer Item in Germany led in vitro testing of the antibiotics in nanocarrier form. The antibacterial activity of antibiotics was compared in free form and nanocarrier form. The genotoxic potential of the antibiotics was checked using cultures of human cells. The researchers also looked at the toxicity of the antibiotics' nanocarrier forms to human cells. Results of these tests will be needed later for regulatory approval. After several years of research, the most promising antibiotics in nanocarrier form were selected for further studies in animal models. These were the M33 antibiotic coupled to dextran-based nanocarrier, the AA139 peptide encapsulated within the micellar nanocarrier, and AA139 coupled with the dextran-based nanocarrier. The SIC Biomayone Research Centre in Spain studied the deposition of inhaled antibiotics in the lungs of rats. The team radio-labelled the antibiotics in free and nanocarrier forms and administered them to rats by aerosol, a nebulizer to generate inhalable aerosols was designed for this purpose by the Spanish company Ingeniatrix. Researchers at SIC Biomayune were able to visualize how much time it took for the antibiotics to reach the lungs and what quantity penetrated into the lungs. Their experiments showed that the AA139 stayed 2 hours longer in the lungs when encapsulated within a micellular nanoparticle, thereby doubling the actual time the AA139 antibiotic stayed on the site of infection. The Erasmus University Medical Center in the Netherlands carried experiments to test the therapeutic effects of the antibiotics in nanocarrier forms. They used rats with pneumonia caused by antibiotic-resistant strains of Klebsiella pneumoniae. The researchers treated the infected rats by aerogenic administration of AA139 and M33 in free form or in nanocarrier forms. They found that association of antibiotics with nanocarriers resulted in increased tolerability of the antibiotic in rats. Regarding the therapeutic efficacy, AA139 dextran and AA139 micelle appeared most promising, showing increased survival of infected rats. After 4 years of research, the PneumoNP team delivered a functional diagnostics kit and showed the added value of new antibiotics coupled with nanoparticles in the treatment of respiratory tract infections caused by antibiotic-resistant bacteria. The research is paving the way for the production and use of safer and more efficient antibiotics that will cure patients like Martin.